So, good morning, all participants, and a warm welcome from Avantor and Leica. Happy to see you there. Before we start, and before I will hand over to Falco and Patrice, I will give you some housekeeping notes for that webinar about how to maximize the outcome of your 3D cell cultures. Don't worry, no need to write something down. We will send you the recordings and all materials of this webinar right after the webinar. So you will get all the stuff in your email box. So no need to write something down in parallel. Questions. We appreciate every question you might raise during that webinar, and don't hesitate to do so. If you want to raise a question, go in the question area. So where the question mark, this little icon on the top of the screen, you just choose that one and raise your question there. We will make sure that you will get the answer right in time and the correct answer in time. And if there is something left after the webinar to be answered on, we will get in contact with you guys after the webinar and clarify any questions we can't answer during that webinar. And of course, we will get, or you will get, a certificate of attendance of that webinar. We know that this is important, especially if you are doing some pharmaceutical investigations. You need a webinar certificate for that. So we are happy to provide that to you as well. Any questions left after the webinar? No problem with that as well. Just write them to webinar@avantorsciences.com. We are happy to answer, and we are happy to hear with the colleagues of Leica. And having said this, I hand over to Leica, and thank you for giving us that webinar today. I'm sure you will give us much of insights about how to maximize and unlock the full potential of your city cultures experience during the webinar. And welcome, Falco, and may you introduce yourself a little bit to the audience. Thank you.
All right. Thank you very much. And also welcome from our side and from my side. Thank you for taking the time today that we can speak a little bit about 3D cell culture experiments. My name is Falco Krüger. I will briefly introduce your speakers of today. So I'm a plant cell biologist by training. I did my PhD in Heidelberg and started in 2018 with Leica Microsystems. With me today is also Patrice Mascalci, who is also has a PhD in cell biology and microscopy, and is not only a microscopy expert, but also really expert in advanced image analysis and programming. And this is also why he is today sharing insights of AVIA and how you analyze 3D cell cultures, particularly organoids. That being said, what are we looking at today, or what are you about to see today in this webinar? So unfortunately, we don't have time to cover all of these solutions that you see here displayed on the screen. I will focus about 3 main pillars that are relevant to, in the end, create an entire cell culture experiment. And this starts, of course, with growing your cell culture or nurturing your cell culture until you get to the point where you, in fact, do your experiment. And for the experiment or for the documentation of the experiment, you need a research microscope. And for this, I'm going to introduce the Thunder imagers to you. And once this is done, Patrice will take over and finish the experiment, not with a nice image, but with a data set that is then being analyzed. This is, of course, also for you, usually the end of the experiment and not just creating a fantastic image. Very important, but it's not the end of the road. So this is what we are focusing on. The other microscopes or solutions that you see here, so for example, the M125C, for example, would be an example of a stereo microscope. This is not being covered today. However, there are also other resources that you can find on our homepage at leicamicrosystems.com, including also the Mica MicroHub and the Stellaris platform. So if you are interested, of course, you can always reach out to us or go via our website, as mentioned earlier. So when we are talking about cell culture experiments, usually what people tend to forget is all the work that goes into the cell culture before you even do the experiment. And this is already rather, maybe not complex, but requires a very high quality of your daily work. and proficiency that you need to apply to have reliable and robust cell culture experiments. And right now we are looking here just at a very broad workflow that covers 2D cell cultures. So where you start with seeding cells, and these cells, of course, need to grow over time, and you exchange medium, keep them happy, alive, and without fungi or any bacteria. And even before the experiment, or let's say before to identify the perfect timing for an experiment, you need to do also confluency checks, right? This is important to have also reliable and robust results during your scientific experiments. But of course, confluency checks are also relevant and very important when you want to decide whether to split or not split cells during the nurturing process. All of these individual steps can and should, of course, be documented, and the data that is generated there also needs to be handled. So this has a few implications, could also include a few pain points, but it was very important here for you and for everyone who is doing cell culture experiments is, of course, you need to record an image of the cells that you are growing. And usually what people are using is the so-called phase contrast. This allows you also to create a very nice contrast when cells are not labeled with dyes or fluorophores, where you would then also need, of course, fluorescent light excitation and a detection system. Here you are simply using transmitted light contrast. This is the phase contrast. But when we are talking about cell confluency checks, this is of course not the basis for your cell confluency. What you want to do is really to measure the area where the cells are growing versus the area of the cells or where there are no cells. And this, as I mentioned earlier, can include a few pain points. First of all, in standard life science research laboratories, doesn't matter whether it's basic research or whether you're working in pharma, there are a lot of different users handling cell culture, right? Usually there are a limited number of hoods, and there's like a nice schedule where everyone then can transfer and nurture their cell culture. So a lot of different people are using usually a small amount of instruments, and this includes also the documentation system. So here, what we have experienced in the past is that the software interface is usually, especially with older systems, not the most user-friendly one. And due to a certain turnover in the lab, this might be rotating students, master students, or PhD students, there's a lot of knowledge drain usually when it comes to proper maintenance of cell cultures. And so not every member in the lab who has to do cell cultures also experienced to do the required documentation. And this includes, but is of course not limited to, how to adjust light intensities, how to adjust the imaging system that you're using in your cell culture to create repeatedly the same image quality, but simply the same data that you afterwards would quantify out of that. So repeating imaging over weeks and months, right, that you can always document this in your lab book, for example, requires a certain expertise. And then a very important point when it comes to a clean work in your bench, in your clean bench, that's why it's called like this, is that most of the systems are using USB keys to transport data from away from the system, which is, if we just think about the USB stick that you have in your pocket of your pants, simply means that, yeah, this is a source of contamination. And this is something that you would usually, not usually, you would really forbid to use in a cell culture hood. So as a last point, when it comes to cell culture experiments and the preparation of the cell culture toward the experiment, is the way of how confluency is measured. Confluency, as I said earlier, is the area of the cells versus the area where there are no cells growing in the field of view or in your well or in your culture dish, for example. And sometimes people also tend to simply do like a rough estimate, like a gut feeling whether or not this looks like 80% confluency or this is 60% confluency. However, most of the time there are no reliable and objective measures of confluency checks. And this is where MaTOTL comes into play, because it offers not only a very intuitive and user-friendly software interface, it also uses a single criterion to measure cell confluency. For you as a user and for the entire user group, there will only be in the software interface, or there is only one single slider that says confluency and then sensitivity, and you mark or move a slider. You can also type in a number, like for example, any number between 0 and 100. And this is your standard measure for your confluency checks. If you then in the lab agree that we always measure our confluency at a level of 75, then this is your standard measurement level and everyone can use it. The software interface then also guides experienced and inexperienced user to optimize the imaging parameter. This is done in a semi-automatic way where the system actually guides you and has like small checkboxes to guide you through the acquisition of an optimized image. A very important feature that I would like to point out with MaTOTL is actually that you are able to wirelessly transfer images, either directly to your smartphone or tablet or to any receiving device. This is done by creating a small hotspot, and with this you can transfer data. This is really cool because you're not required to introduce a contamination hazard into your clean lab bench or hood. And this is very important. Of course, it's also possible to reproduce imaging parameters if you have found them in the past and you know them to be the most optimized settings. You can also, for example, reload old or saved imaging parameters. So this altogether creates, of course, a very nice package that MaTOTL offers for your everyday cell culture work. And here you can actually see also that it fits nicely into a clean bench or here into a hood where you can then do your cell culture experiments, but also the routine cell culture checks. And this is what is done right here, where you simply put the 6-well plate onto the surface of MaTOTL and then do your transmitted light imaging. When we are talking about 3D cell culture experiments, and here particularly in growing organoids, this is a completely different level when it comes to complexity already of the cell culture growth. We are not only talking about, okay, I need to seed cells and they need to attach and then they start dividing and growing and are happy. Growing organoids is a science for itself. Okay, and here I also don't want to give you any particular advices. I simply want to sketch a little bit the workflow that is required to do organoid growth in or as a cell culture experiment. So depending on what kind of organoids you're growing, you either have to use tissue sampling or genetic engineering, right? Whether you want to grow organoids from a cell culture or you maybe have patient-derived tissues. Then you need to isolate cells and let small organoids grow, or you need to wait until the embryonic bodies are aggregating. And over the course of days, weeks, and even months, depending on how big you want to grow the organoid, you have to do, of course, the we would call it the expansion phase. You would let the organoids grow. Sometimes you also start with a group of organoids, which then are later on isolated. This is also where MaTOTL can, of course, offer you a nice access to your cell culture. So during the course of the growth, you can always quickly check, put your well plate underneath MaTOTL and then record an image and immediately go back to the growth chamber, or you can then also use this for documentation of what kind of organoids are you going to pick for your later scientific experiments. And this is also what I want to point out here. So it's not only MaTOTL can not only be used for 2D cell culture, but also 3D cell culture. It, of course, offers you a live image, so you can always do real-time monitoring of your organoids, and you don't need to touch or manipulate the organoids in order to image them. You can also image them through the thick plastic and matrigel layers that you might have in order to record the current state of the organoid. And this is also where we are now slowly moving into the realm of the scientific experiment and imaging of organoids. MATIOTL is not only an isolated system, it can also be used, of course, in a workflow together with our other imaging solutions that we offer. And the Thunder imagers would be the next one that I'm going to talk about. So just as a small wrap up, MATIOTL is a very intuitive and user-friendly system, which helps also inexperienced users to have a reliable and robust documentation of their cell culture. All right, now let's go to the part where we are actually doing the scientific experiments and later on also the image analysis. So we start with our cell culture and then we skip forward maybe 50 days or 80 days, depending, as I said, how big you want to grow the organoids. And then you do your experiments. People are using organoids not just because it's so nice and complicated to grow them. No, organoids are simply more relevant, physiologically relevant compared to a cell culture. Because usually biology is a three-dimensional matter. So if you can grow in different cell types in a three-dimensional context, also their interaction is more physiologically correct than in a single cell culture. And that's why the entire field of cell biology or also medicine is moving also toward these models, because as I told you earlier, you can extract also from patient-derived tissue, you can extract and grow organoids. And this is something that is very important when it comes to individualized medicine. And for this, at some point, you also need to check whether or not a certain cell type, for example, is growing in your organoid. You want to, I don't know, maybe find out how the interaction, how the development of individual cell types is being or is happening in your organoid. And for this, you need to do fluorescent imaging. And usually what people are using are confocal microscopes, which is perfectly fine. I don't want to say anything against it. However, what I would like to offer you today is a different solution that deals with a very big problem, why people are in fact using confocal microscopes, and this is the out of focus blur. And the out of focus blur is what usually keeps the scientist from using a fast and very sensitive detection system, so an epifluorescent or wide field microscope, because it always have tons of out of focus blur. And as you can see here, you are not able to distinguish any particular structures or information inside of your, here in this case, organoid cluster. So what Thunder imagers offer, however, is the removal of this out of focus blur in real time. Okay, so you are still imaging with, and this is where I go next here, you are still imaging with a camera-based wide field system. You keep the speed and sensitivity of camera-based imaging. However, the image quality in terms of contrast is much closer to the image quality of that of a confocal microscope. So what you gain here is actually not only 2D but also 3D information of your organoid in this case. And this allows you to also come to conclusions without going to, in comparison, rather timely or time intense imaging method, what is the confocal. So here we actually closed a gap by introducing those Thunder imagers. And this is also, from our standpoint, a new class of instruments, which is intended to close a long-standing gap, actually. So where wide field microscope in the past have only been used for 2D cell culture experiments and confocals for 3D ones, and the Thunder imagers are actually now able to do both types of application. And a Thunder imager is not only a single system. We offer a family of Thunder imagers, starting on the top left with the Thunder imager model organism, which is based on a stereo microscope. Stereo microscopes offer you to have a very long distance between the observing objective and your specimen, which allows you to, for example, pick organoids and move them from one well to the other, isolate them into a different cell culture. So this kind of large overview, large field of view imaging can be done with a model organism thunder imager. However, of course, since it's based on a stereo microscope, the resolution is of course not comparable to that of compound microscopes. Talking about which, we also have the thunder imager 3D tissue, which allows you to use also stages with up to 8 slides here, where you can have your already sectioned tissues, also rather thick samples, several microns thick, or even 50 to 100 micrometer thick sections can be observed with thunder imagers of the specimen 3D tissue. A very unique system is the thunder imager LMD, which combines not only the 2D and 3D imaging of samples, but also LMD is called a laser microdissection system, offers you a solution when your workflow also integrates downstream analysis of DNA or protein components in your sample. So with that system, you're actually able to cut out relevant areas in your tissue, or even select individuals or cut out individual groups of cells and then subject them to proteome analysis, for example. This you can do with a LMD thunder imager. And then we have the class of the inverted thunder imagers, which start here at the 3D assay, which allow you to do quick assays in 2D or 3D. You can do long-term live cell experiments with a thunder imager live cell, and last but not least, the thunder imager nano, which is a thunder imager that you can integrate into a workflow where you want to prepare your specimen for electron microscopy afterwards. So you can, we have here also a very dedicated shuttle system that allows you to then also go into downstream sample preparation for, as I said, electron microscopy. Okay, big class, but what is actually here the benefit? I told you earlier, wide field systems are usually limited to 2D cell culture, because when it gets more three-dimensional, you add more out of focus blur, and this then hides actually the information that you want to see. Thunder imagers, not only based on the individual classes that we offer, but in general offers access to 3D information in your specimen, including also spheroids and organoids. And this is the main topic, of course, of today. Here, I simply brought an image of a spheroid, so a rather also big specimen that allows you here, instead of having the image that you would record with a state-of-the-art microscope, and here compared to a thunder imager where you have access to all these small cell clusters and structures that you see here, where the out of focus blur is removed. Now, how is it possible that we have both images present, so the blurred image and the thunder image? Well, this is because thunder, and this is a very big advantage, at least in my opinion, offers you to always keep the raw data, because, as I said, you keep the sensitivity and image acquisition speed of a standard state-of-the-art microscope, and this, of course, records the raw data. And at the same time, in real time, the thunder imagers produce also the so-called thunder image. And you can always put them side by side and compare the raw data with the thunder data. You can always go back, okay? And this is something that many other systems in the field actually do not offer, because the raw data is either not accessible or dispersed beyond recognition, let's say. And here now, let's have a look at a very nice example that I recorded a few years ago in Erlangen, together with a PhD student at the time. This is an overview of a brain organoid. And this is basically derived from human skin tissue and then was grown into brain organoids. And what you see here is actually a really big, over a millimeter across organoid cluster. Let's put it like this. And this, in the beginning, we recorded here an overview. this is a tile scan recorded with a 10 times objective, where you can already specify individual cells and also types of cells down to a level of 285 microns inside of the brain organoid. What you can see here, and this is of course scientifically relevant, is that there are areas where there's not so much proliferation going on, but then also here, for example, this region displays already a site where there's more complex tissue organization happening inside of that organoid. And the entire image acquisition, so this organoid here has 4 channels, and as I said, it's a tile scan. The entire image acquisition and processing, three-dimensional processing that we do here, which is called deconvolution, took over a little bit over 3 minutes. So also the speed with which you can record this kind of data is drastically increased. We afterwards switched to a different magnification. In this case here, this is a 20 times long working distance objective that allows us also to move to image through the thick plastic bottom of a multi-well dish, for example. And here, what you can see are really nice distributions of individual cell types. I'm not going into detail what kind of stuff these cells are doing, but what you can see here is the big formation of a complex tissue structure, and then also here other small sites where this is already starting. So this kind of image quality, again, was not recorded with a confocal microscope, but with a Thunder imager. So also here, again, we imaged down to a depth of 100 micrometers, again, for channels, and overall imaging or image acquisition and processing took a little bit more than 5 minutes. And this is, I think, a very fast access to this very detailed information. And from there, you can then also go, of course, and analyze your data. With organoids, this is not the end of stuff that you can do with Thunder imagers. What I also brought is an example from a PI at that time working in Munich. She is working with cleared organs. And in this case here, this is a piece from a mouse intestine, again, also cleared, but you see here the dimension of this piece of tissue, which is several millimeters laterally, but also down when it comes to thickness, several millimeters thick. And what was very interesting here in yellow, you see, I think, a microvilli of the intestine, and then here a cluster of lymph nodes. And the purple labeling is actually not the lymph node itself, but the blood vessels providing nurturing for the lymph nodes. And you can see here that it's really indeed a cluster that is attached to this intestine piece. And this we can then also have a closer look at in 3D, as I said, and we are here going down to 1 millimeter into this clear tissue. Just as an example of, you can really image also bigger three-dimensional structures, not only exclusively organoids, but also other tissues. All right. But what is Thunder Imager? What is the technology behind this? And I want to spend a few minutes here to simply tell you, okay, what is going on? What do you need to understand as a take-home message is that Thunder is computationally clearing. When it's done in real time, as I said, you are recording your raw data as always with your microscope. And the Thunder Imager offers you then either in real time, so instant computationally cleared, or also as a post-processing mechanism, you can only do the computational clearing later, access to three-dimensional information without the autofocus blur. And computational clearing, as the name says, is an optodigital method. This means we are taking the optical properties of the objective and a real-time processing approach in order to come up with this technology. This is something that we at Leica developed a few years ago and launched this imaging systems, I think, end of 2018 as well. And the good thing is it offers you autofocus blur-free images in real time. This works in 2D. Okay, here comparison between the raw data and the thunder data, and also in 3D, because if you collect three-dimensional acquisition of images, which is called a Z-stack, then of course each individual image is being cleared, and this then comes up also and gives you three-dimensional data access. So that's very important to keep in mind. It's in general a 2D method, and it is based on the optical performance of the objective that you are using. So the better the objective, the better is the computational clearing that thunder imagers offer. And if you have lower magnification, a bigger field of view, less resolution, you still get a decent image as you have seen beforehand with a big organoid cluster or the cleared intestine. But what is really happening on the image side? And to explain you on a, let's say, I hope at least on an understandable level, I would like to talk a little bit about what your image that you are recording with a standard wide field microscope is actually composed of. Of course, you have here as an orange line your signal of interest. Okay, that's a given. You have labeled a certain, I don't know, group of cells, right? And these cells you are then exciting, or the fluorophores that are expressed in there, you are exciting, and then you want to record the image of this. However, your camera, for example, also has a certain what we call the detector dark current. So there is a certain level of intensity that is being detected also when there is no signal coming from the specimen. Then also a very nasty portion of your image is autofluorescence. Depending on the thickness of the tissue or what kind of tissue you are looking at, the autofluorescence might be stronger or less strong. Yeah, as I said, it depends on the density of the cells in the tissue. A liver, for example, is one of the rather problematic tissue types. And this is also contributing to the signal that you detect when you record an image of your 3D specimen. Then you have, and this is most important and becomes more relevant if the sample gets three-dimensional, as it is for organoids the case, the out of focus signal, because your wide field microscope uses a certain objective, and this objective has a so-called focus plane. And this focus plane, or what focus plane means, is that in this volume, the image is the sharpest. Okay, but of course, there's also light being detected from above and below the focus plane, and this light is being detected and hides, and this is the actual out of focus blur that is then hiding the structures or information that is in the focal plane. And this can be dependent on the thickness of the 3D cell culture that you are using, can be more or less, right? And all of these components, which have in fact nothing to do with your signal of interest, also contribute to the image that you are recording. Let me give you an example in a second, simply here describing what thunder imagers do. They remove the component in the image that is not related to your signal of interest. I have to make a small side note, for example, also the detector dark current, so the signal that is inherent with the detection, the camera chip in the end that you are using, this is also not being removed from the image, simply to have here then also a proper ground level of your intensity. Okay, but everything else, the out of focus signal, the autofluorescence, all of these kind of unwanted components of your image are being removed, leaving you with the signal of interest. In reality, this can look something like this. So here we have a very, or rather flat sample, which has also here a little bit, as you can see in the center of the image, the background blur is increasing. And hopefully what you can also distinguish is that there are a lot of individual cells being labeled. Okay. The top image is the raw image that we have used, and the image below is the thunder image. And similar to this schematic overview that you can see here on the left-hand side, where there is along the screen line of interest, we have recorded here the intensities. And what you can see is that in the individual spikes, yeah, they are visible, but depending on what amount of unwanted signal is actually in the image, they are getting less and less distinguishable. However, maybe you want to quantify these kind of nuclei that are labeled here in that case. If you would now apply a thresholding based on intensity, maybe here at the level of 1400, you would actually lose specificity here with these spikes, because the signal here on top would be simply counted as one big blob. Okay, and this is where thunder imagers come in handy, because we remove this out of focus blur and keep the individual spikes and also their relative intensity differences here accessible for you as a scientist then later on to analyze. And this, as I said, works in 2D, but also in 3D. Here, a live fly-through through the raw data set compared to the Thunder data set. So, as I said, it's a 2D method that works, of course, also in 3D when you record several 2D images along the optical axis. So this explains, hopefully, really the power of Thunder imagers, giving you access to three-dimensional information in three-dimensional cell culture. Okay, for a second, I only go a little bit more technical, simply to explain to you a difference between general ways of improving images in a wide field microscope, because most of you, I bet, will have heard already about deconvolution. And you might wondering now, okay, deconvolution, yeah, usually deconvolved images looks sharper and more crisp. Is Thunder nothing else than deconvolution with a fancy name? No, it's not. So when you want to improve images in the wide field microscope, there are two basic approaches. One is an exclusive approach, where you remove information or intensity or components from the image. And then there's the inclusive improvement, where you are not removing information, but you are repositioning information. I show you the difference. Here, what you can see is something that we call a point spread function along the optical axis. So here, the distribution of light of a sphere-like structure, simply here distorted due to the optical system that we use for recording this glowing sphere. And an exclusive improvement would mean that we remove these out of focus information from the image. Okay, and this exclusive improvement is something that computational clearing does. This exclusive improvement is, by the way, also something that the confocal microscope does. But a confocal microscope does this with a physical pinhole, okay, where then the out of focus light is really blocked. For a confocal, just to be here clear, the point spread function would also look a little bit different than compared to the Thunder image. However, the general mode of improvement is comparable. Now, when we talk about deconvolution, right? The question is Thunder simply deconvolution? Deconvolution is an inclusive improvement, meaning the out of focus light is based on a physical formula back calculated or repositioned into its point of origin, because the distribution of this three-dimensional signal is more or less quantifiable or measurable, because it mainly depends on the lenses that we are using, and that's why you can also do a so-called image restoration. So you can see the difference, right? Exclusive versus inclusive improvement. Both have their own advantages and disadvantages, and you might wondering, okay, so Thunder imagers do here this exclusive improvement, but I also like here the improvement that I see with deconvolution. Is it also possible to combine both of those modes? And yes, this is possible. And here as a last example, I would like to show you really what is the power with Thunder imagers that we offer, combining Thunder, so the computational clearing, with the 3D deconvolution. And you can see here the raw image of a bacterial culture. Okay, those are Salmonella expressing a cytoplasmic GFP. And this was, by the time I took the image, a live cell culture where I recorded a volume scan in the specific well of this well plate, and I simply wanted to see what is the maximum that I can here detect. The experiment, to explain to you briefly, was like the following. So the cells are expressing cytoplasmic GFP, and in the medium there is a peptide, a red peptide, a red fluorescent peptide, where the question is, does this peptide it's known that it attaches to the cell wall of the salmonella, but is the peptide actually able to enter the salmonella? In the end, here it's simply a dye that we are using to see this, but it has implementation when it comes to antibiotics, right? So is this component able to cross the cell wall of the salmonella? And usually the scientists were using confocal microscopes or even super resolving image acquisitions. However, or to solve the question. However, with the Thunder imager, as you can see, we could clear a lot of the out of focus blur and background from the culture and get access to individual salmonella bacteria. And we could also zoom in. So this image here, I've simply now cut out a few examples, and one I would like to show here. This here is a single salmonella extracted from this data set. And you can see here the individual modes that I have used. On the left-hand side, you see the raw image, the green channel, red channel, and the overlay. The ICC image, this is instant computational clearing, so the Thunder image, basically looks the same. Why is this? It's very simple, because the bacterium that we are looking at has a certain dimension, and the focus volume of the objective that we are using, in this case a 100 times objective, is bigger than the bacterium itself, and that's why you see the entire, here in this case rather blurred structure in this field of view. Okay, but we recorded a 3-dimensional Z-stack. Okay, so we have, beside this individual focus plane here that I have extracted, we also have information above and below. So what large volume computational clearing does, so LVCC, is the combination of clearing out of focus blur and restoring the image. So this inclusive improvement, meaning light that has been scattered in 3D is calculated back into its point of origin. And you see here the drastic image improvement, also information improvement that you get out of this. You can even start seeing here substructures in the bacterium. So as a result of the experiment, you can actually state that this peptide is not entering the cytoplasm of the salmonella. It's simply here due to the lack of resolution that we could not distinguish this in this experiment. So I know that this is not a 3-dimensional cell culture. I just want to show you that this here is, let's say, the resolution maximum that you can get out of a Thunder imager. And this is pretty impressive for your everyday live cell experiments. Of course, this can also be applied to 3-dimensional tissue, as I've mentioned earlier, where here the experiment is RNA fish, so fluorescence in situ hybridization, where you want to highlight individual RNAs in this case that you would like to image and then also quantify. So you want to count the dots, which is impossible in this image over here. Still not very robust in terms of quantification when we are talking about this instant computationally cleared image. But combining again Thunder with 3-dimensional deconvolution, you get access to quantifiable information. And when we are talking about quantifiable information, I want to now close the circle and go back to our introduction where we had this out of focus blur. And now we are looking here at an organoid cluster. But as I said, a nice image is not the end of your experiment. We are now want to entering the realm of how can we quantify this data? How can we analyze information out of this image that we have recorded? And this is where I hand over to my colleague, Patrice.
Thank you, Falco. So hello everyone, I am Patrice Mascalchi, EMEA Sales and Application Specialist for Avia Software. And today I will present with some slides and a live demo this solution that we offer in our portfolio at Leica Microsystems. So the EVIA solution comes from a team that has been working on AI projects, AI for image analysis a long time ago in 1999. And in 2017, they've been deciding to prepare a package of applications and tools that has been named EVIA. And then in 2021, this team and the product has been joining Leica Microsystems. As you can see here with the different versions from 5 to 13, a lot of different features that have been added over time, and it is a continuing process on pushing for AI related tools and improvements that we can do with the software. So the goal for this software is really to take care of what AI can offer to the world for of image analysis, of course. And the goal, of course, with these AI tools is to make them accessible for all, because you might not have skills to code, to program yourself with some command lines. So that's why here in EVIA everything is prepared so that it's only a user interface with buttons and some values that you can enter, so that you can have access to all these tools. The main, of course, application is that the detection of objects, cells, vesicles, nuclei will be simplified with these technologies. And of course, because I already introduced the fact that AI is relying on some deep coding in the background, it's really important here for you to be able to have a single platform that can handle a lot of these different steps. Before I continue in the presentation, I just want to remind some definitions for the different keywords, such as artificial intelligence, which is a big group of different tools, but we also have then a subgroup, which is machine learning, where we have the ability to make our computer to learn something from our input. And then a subgroup again of this one is deep learning. So you probably heard about deep learning keywords, because of the autonomous cars, because of also a computer being able to beat chess players, for instance. So all of this means that the deeper we go into this subcategory, the more we can achieve, but maybe also the more it is restricted to some applications. So that's why it's quite interesting to have still complete access to different levels of technologies with AI. Here I am presenting this slide common workflow or pipeline that you would do for image analysis, for instance, where of course you have on the left an image which has some, let's say, some difficulties for the computer to recognize what are the shapes of interest. You with your eyes would visually see something that you want to detect, but the computer needs to match that. And in order to match this is where the pre-processing step here, where we can see there is a clear map then, which is identified about the objects of interest, can benefit from machine learning or deep learning technologies. Again, here, the ability to potentially train this AI to recognize some shapes is really also very interesting at that step. Then the next step will be that we are generating some objects. We can color code them. We can then extract some measurements as the next step. But again, then there is potentially the fact that you want to decide on, do they belong to some groups, some phenotypes. And this is also something that can be, let's say, powered by AI. And then you will export your results. A last tool that we are offering in EVIA is called ATL, for EVIA that learns, and it is actually a prediction of the parameters for the analysis at that step here in the middle. Why do we benefit from this? It's because, for instance, a very critical and simple parameter we have to provide is what is the minimum size of our objects and what is the maximum size. This prediction, this deep learning based prediction, will take care of this. So Now I want to give a quick live demonstration about what we can do with machine learning for the segmentation, so for the detection of some objects. And I will switch now to the software for live demo. So our sample is an organoid cluster. Here we can see that this data is in 3D. I can navigate here in 3D. And what I will do is to use this machine learning pixel classifier interface that I have on the right. And to do so, I will then go to the 2D view so that I can provide precisely some shapes that are of interest in my detection. If I zoom in, we can see that, and you will recognize that in your images as well, you have some similar, let's say, difficulties, such as the staining is not plain, and you will have to still to detect something which is plain. So to do so, what we can do is to provide an example of what is background here. And on top of this, we can provide an example of what would be a positive match in this context. So this machine learning technology will learn from this context. And as you can see here, I can adjust the brush of my drawing tool here to provide examples and that would correspond to my objects of interest. I can also provide the fact that this blurry region here should not be detected as positive. I can just mark it like this. Once I've done this, here for the demonstration, I will activate a region of interest that I've been preparing, and I can just activate this region, press the teach, which is just making the software learn, and apply. And as you can see, in a quick way, we can obtain a map of our, let's say, objects of interest, and we can then have this effect in the 3D environment. Now, if we want to have a detection as objects, so which would be the second step, here we were doing some pre-processing. I can select my map, my nuclei map as an output of my machine learning, and here press the start button to generate some results. I will then switch to the 3D environment again, because now I want to see the effect of this 3D object, 3D nuclei detection in my 3D environment again. So I can click here again on that button here for the 3D. We can have again our view of the 3D image, input image, and on top, in the region that I've been defining, we will see now that we will have the result displayed. So displayed as color coded, as you can see. And now we have the ability to extract measurements out of these objects, plus also interact directly with these objects if we want to. If we extrapolate this to the full image, we can see an example here of a full map. As you can see also that depending on how busy is the computer, there is the use of blocks to make it appearing over time. And we can see that on top of this, then we are able to detect all the objects in our image. Now coming back to the presentation, I was mentioning that we also have the access to deep learning technologies for the same goal of identifying objects. In the software, we have the ability to even apply directly some pre-trained deep learning tool. And that's a good benefit because here we can see that if we compare in the software area the non-AI detection of some neuronal cells, so the soma of the neuronal cells, here we would miss quite some cells But this AI-powered soma detection here is really filling the gap and allowing you to have precise detection and a better detection of these cells, even if it's in a dense environment, as you can see in the image. And then I've been citing the fact that we are also providing AI to predict parameters. And here is an example, for instance, from this image, you could, in a single click, have access to this type of result with the prediction again on what is the minimum, maximum thresholding on the image that you would do manually otherwise. But what is interesting is that you can still access the manual parameters afterwards. And finally, I want to cite the fact that we sometimes want to go beyond just the simple detection of objects and potentially go through more insight into the 3D environment and interactions between objects. And that's why we have the availability in the software to have a multi-compartment and multi-scale, as you can see here, tool to detect multiple types of objects. So here we have different results, different examples, but coming from the same tool. And with the color code here, for instance, of objects depending on the distance to the nucleus, for instance, here on the top left. I will finish with the fact that in this solution, it's not only about providing a good AI tools, it's also providing a good interactivity for the user to find back some resources which are important to understand what to do in the software. And this is why we have a search bar at the top of the software where you can access YouTube tutorial videos. You can also access online documentation. And this comes at the top of course, a training which is provided by application specialists and customer support. I will finish with this nice image, nice video of a Thunder, let's say, volume which has been acquired, and where you will see again some results that were obtained with Avia. Thank you for watching, and I will be able to welcome some questions, answer some questions now. Thank you.
Yeah, thank you, Patrice, to giving us all that insights and Falco as well. I received a couple of questions as I see time is running quite fast right now. The first question I received, Patrice and Falco, is it possible to classify different cell populations in a tissue? Please answer on that if you, I think. Yeah,
so yes,
sorry.
Thank you. It's both, indeed, it's both the preparation on the staining, of course. You cannot expect to classify cells without providing some information, or maybe it's possible to classify on the shape. And indeed, you could go to the software, to the analysis software, and use all these, let's say, distinctions, these features in the software to provide then the classification parameters. But indeed, providing one parameter is something we usually do, like for instance, small cells versus large cells. Whenever it comes to multiple parameters, so n parameters, there we can use machine learning technology to make the computer learn from examples that you provide all these parameters in one go. And that's really one key benefit indeed of the software.
I mean, Patrice, I think what you could also do is then that you combine individual properties of cells, right? I mean, a classic way of differentiating different cell populations would be with different markers, right? And you could, or I don't
Exactly.
know, would it, right? So you have different colors, but it might also be that the presence of two colors or two markers in one cell could also lead to a very specific profile, let's say, of a third.
Yes, exactly.
Cell type.
The example I provided in the live demo today was on a single color, but you can have multi-channel inputs for the same tool, which is a lot in line with what you said, so helping a lot on the phenotyping.
Okay, and let's take just a minute for our next question. Yeah, and this question is about what other cell culture models can Matteo TL be used for? General one, I guess.
Yeah, As I said, so as long as you can grow it in a multi-well plate or in a cell culture dish, or even on those cell culture microchips, you can look at them, right? So you can also put a cell culture flask underneath. So on this stage of Matteo, so the images that you have seen of Matteo today are basically the version where you simply have a flat stage surface and then the opening where you then would do your transmitted light imaging through. But of course, what we would be able to offer is a stage device where you can then place directly your well plate in and then have a small stick to navigate or joystick to navigate your sample over the area of the stage. So this would be a more controlled way if you want. But other than this, as I said, imaging dishes, multi-well plates, slides, whatever, right? You name it. So the standard cell culture specimen holders are possible. Yeah.
Perfect. Thanks, Falco. And seeing that we are at time right now, so but don't worry, we will take care about every question raised in the question area anyhow. So I will do the outro. As mentioned in the intro, you will get a follow-up mail from Avantor sharing the recorded session. You are happy to share that with your colleagues as well. And and if you have any questions left, then please don't hesitate to raise them, as said before, to webinar@avantosciences.com. We will take care about them, sending that to the Leica specialist and an Avanto contact for you to getting your issues and problems solved or your questions solved. Having said that, I'd like to thank you, first of all, the participants for joining us today. So thank you for joining. And Falco and Patrice for giving us all that amazing insights about how to optimize and make the most out of the 3D cell cultures in your everyday work. Thank you, and I will close this session right now. Goodbye.
Thank you. Bye-bye.
Thank you.